Okay, I realize I forgot to introduce myself at the start of the session. So while we're doing slide changeover, I'm Paul Diggard. I'm a virologist based at the Roslin in University of Edinburgh. My co-chair, Caitlin Greenland-Buse, is at the University of Liverpool School of Tropical Medicine. And I'm passing over to Caitlin to introduce the next speaker.
Oh, hi everyone. I'm excited to introduce our next speaker, which is Miss Yeonjae Lee, master's student from the Pirbright Institute, and she's going to be talking to us about Sarbecovirus usage of mammalian ACE2, investigating the receptor binding and genetic determinants of host range.
Thank you, Caitlin. I'm Yeonjae Lee. I'm a master's student at Imperial College, and today I'm going to be talking about my master's project that I've been doing for the past 6 months at the Pirbright Institute. But before that, I would like to thank UKICM for giving me an opportunity to have my first ever talk at conference today. So my project was on investigating the Sarbecovirus host range and receptor genetic determinants of their receptor binding. As we are all aware, SARS-CoV-2 spike protein entry is determined initially by the spike protein and host ACE2 receptor protein interaction. And given that SARS-CoV-2 is presumed to be originated in bats, bats could potentially be the route to human spillover transmission. And going further, spillback transmission from humans to wildlife and domestic animals is also a major concern. This heat map illustrates the receptor usage profile of SARS-CoV-2 and SARS-CoV. And as you can see, both of these viruses have a broad host tropism, and hence there are high possibilities that there are spillover potential from human to broader animal species besides the reported cases. So based on these informations, the aim of my project was to investigate the usage of mammalian ACE2 by sarbecoviruses, particularly SARS-CoV-2 VOPs and bat sarbecovirus, and to identify and investigate the sequence determinants of differential binding to ACE2 and also the host reservoirs to prevent future spillover. So after the emergence of SARS-CoV-1, there has been continuous discovery of sarbecoviruses from various animal species. And recently, there have been many new sarbecovirus identified from bats belonging to Rhinolophidae family. Before I explain which sarbecovirus were used in my project, I would like to point out that we particularly focused on receptor binding domain of the spike, as that's where most ACE2 contact residues are located at. So if you look at the heat map, this illustrates the amino acid identity of sarbecoviruses compared to SARS-CoV-2 wild type. And as you can see and expect, phylogenetically differently related sarbecoviruses have lower similarities to SARS-CoV-2 wild type. And so based on this information for my project, we particularly chose bat sarbecoviruses that have high, medium, and low similarities with the wild type. So those are these three banals. They have high similarities with just a single substitution at position 498 and an additional mismatch at 493 only for banal 236. And we also had RCO309 and REXCS203 with medium and low binding with due to their unique RBD sequence with high amino acid deletion in their ACE2 contact regions. And additionally, we also included WIF1 and RCO309, which are bat sarbecoviruses that have spike proteins that are highly related to SARS-CoV-1 rather than SARS-CoV-2. So to examine their bindings, I've initially cloned RBD and ACE2 ectodomain sequence into protein expression vectors and transfected them into XP29F cells to express some recombinant proteins. And those binding of those recombinant proteins were then quantified using ELISA. We also performed another binding assay, flow cytometry, to screen the binding of the recombinant proteins RBDs and binding of RBD recombinant proteins to ACEs that's displayed on the surface of the cell. And these two different methods were used for confirmation and validation purposes. So upon correlating ELISA and flow cytometry binding data for human ACE2, as you can see, there were SARS-CoV-2 VOCs and most Sarbecovirus RBDs had high binding with human ACE2s, except REC-CS203 and RCO309 expected. And similarly, white-tailed deer and Syrian hamster ACE2s had similar binding patterns to human ACE2s, except there's no binding observed for Delta RBDs to Syrian hamster ACE2s by ELISA. However, we predicted that this is an anomalous data as Syrian hamster ACE2s supported binding of most of these RBDs. But rather than that specific data, we could see the strong agreements between these two methods validating our result. However, this wasn't the case for bat sarbecoviruses. There were differences observed between these two assays, where there were more flow cytometry than ELISA positives. So upon comparing three bat ACE2s with the binding to their IBDs, and also based on previous publications where they say that little brown bat supports entry of SARS-CoV-2 pseudoviruses, we came to a conclusion that flow cytometry is more sensitive than ELISA for detecting these two interactions. So this is our flow cytometry data, and as you can see, SARS-CoV-2 wild type, Delta and XBB.1.5 had similar binding patterns. And among them, Delta bound ACE2 is better than the other two. And based on our bat Sarbecovirus data, as expected, phylogenetically related RS431 and BIF1 had similar binding patterns, whereas RS431 showed that they could use broader ACE2s compared to BIF1. And among banals, banal 236, they had similar binding patterns, obviously, but then banal 236 had bound ACE2s better than the other two. And so by comparing the sequences of Sarbecovirus with stronger binding to the other Sarbecoviruses that had similar binding patterns, for example, 236 to banal 52 and 103, we could analyze that just a few mutations or even a single mutation could enhance ACE2's binding ability. Finally, RCO309 and REC-CS, as expected, showed poor binding to ACE2s, except for RCO309 showing binding with Lee's horseshoe, Chinese rufous, and intermediate horseshoe bat. However, if you've seen NASA's poster, you have noticed it, but the RCO309 only showed entry into Japanese horseshoe bat, which then we could conclude that RCO309 mainly uses Japanese horseshoe bat ACE2s. Whereas for REC-CS203, it failed to show binding with all of our assays. So we came to a conclusion that they don't use ACE2s as an entry receptor. So to conclude everything, Sarbecoviruses have a broad host tropism, and Sarbecoviruses in bats, geographically isolated in Southeast Asia, have a transmission potential to bats all over the world. But also we have seen exceptions to RCO309 and REC-CS203. However, they shouldn't be excluded from having a spillover potential, as just a single mutation in RBD, especially at an ACE2 contact residues, could enhance binding to human ACE2s and ACE2s in general. So I've titled this slide as future work, but it's not my future work anymore because I'm not, my project ended in Purbright Institute, but if I were to, and what's planned with my supervisor is that you could gain a complete set of binding data using the two different experimental systems to get a clear picture of the trends and changes as new VOCs evolve. And also we are planning to include the new subvariant, the BA2.86 in our assays, And finally, correlate the finding of Sarbecovirus host range to affinity and indigeneity, which is currently being done by Joseph Newman and Nasia. I would like to thank my two supervisors, Dallin and Joe, for their support for throughout the 6 months. I'll also like to thank Nas for all the help and the members in the Fulbright Institute. Thank you.
Thank you for a great talk. So open up for questions. Has anyone got any questions?
It's really interesting to see the that sort of change at the end on RCO 319. Is there anything in particular about the bats that were more likely to accommodate that mutation in their environment or in the size of the colonies that there are that would indicate whether or not there is a greater spread in some species than others? That's too much. That's too. I'm just wondering what you found on a micro level. I wonder if there are any environmental explanations.
So from what I know, the clade 2 bat Sarbecovirus mostly in Asia. They don't use ACEs as the entry receptor, but geographically, I'm not sure if that's due to environmental effects or anything like that. Sorry.
Thank you.
Thank you for a great presentation. That was really, really exciting. And you mentioned that flow cytometry and ELISA, when compared, you thought the flow cytometry might be more sensitive than ELISA.
Yeah.
And I just wondered whether you'd considered the way in which the antigen is binding to the ELISA plate and whether that might improve the sensitivity of the ELISA assay compared to the flow.
Oh, yeah. So before our ELISA, we have requantified the proteins to make sure that equal proteins are loaded in the plate. But even by doing that, that's what we came up with our data. So what we think that might have happened is that there's sorry, how do you pronounce? The steric hindrance in ELISA might have affected that, and also because how ACE2s are presented in our flow cytometry is different to ELISA, so we think that might have made that differences.
Thanks very much. Great talk. I've comment terms, perhaps that'll help you, and then a question. So we've developed white-tailed deer cells and organoids, and on HEC293 cells, white-tailed deer ACE2 seems susceptible, but on cells derived from white-tailed deer, it doesn't do anything. So my question for you is, how well does your binding assay correlate with your entry assays?
Oh, I'm not so sure about our entry assays, but I'm pretty sure there are lots of articles about white-tailed deer, SARS-CoV, the VOCs being discovered. I'm not sure. SARS-CoV being found in white-tailed deer, so I'm pretty sure there must be infection.
Maybe I should have. That's my fault. I should have clarified my question. So if you took your heat map of your binding assay, and then you correlate that with your colleague's assay where you're doing entry assays on HEC293 cells, how much of the heat map holds up when you do your entry assays? Or do you have false hits?
No, I'm pretty sure there are entries. They do correlate together where we see binding with the white-tailed deer ACE2s, and also there is an entry with our viruses.
So in the interest of time, I think we'll move on, but thank you again.
Thank you.